Hello everyone, welcome to Vazyme's webinar. A big thank you to the team for making this event possible. My name is Sitar Lee, and I will be your host throughout the event. Now, before we get started, I want to dip into a few things on how to get the most out of today's webinar. First of all, this webinar is being recorded. The on-demand version will be available after the webinar ended. We will send out emails to share the link with you within the next 24 hours. So if you enjoy what you see today and you want to share with your peers or colleagues, you are welcome to do so. If you're facing technical issues, please refresh your browser and wait to establish a new connection. You can also send us a message using the Q&A box. So today's event will run as 3 parts. Firstly, we are joined by Jiaxu Wang, Principal Investigator at the Genome Institute of Singapore, who will start the webinar with the first presentation before handing to Shu Yuanding, Technical Support Engineer at Vazyme. Once the speakers are done for the presentation, we are open for Q&A. So if you have any questions during the event, use the Q&A box on your left-hand side. We will get to them as many as possible. Now I will hand over my time to our first speaker, Jiaxu Wang. Dr. Wang, whenever you're ready.
Hello everyone. Good morning, good afternoon, or good evening. This is Jiaxu from Genome Institute of Singapore. Thanks to Vazyme for giving me the opportunity to share our project here. Today's topic is development of a single-cell RNA structure sequencing approach to study human neurogenesis. As we know, since 1958, Francis Crick published the central dogma and described that DNA could transcribe into RNA and then RNA could translate into protein. And later, more and more studies have shown that there are 70 to 90% of DNA could transcribe into RNA. However, only 1.5% of the RNA could translate into protein. This suggests that RNA may play more roles than we expected. Actually, even for this 1.5% of protein coding gene, the correlation between RNA expression and protein expression is only 0.4, meaning only 40% of the protein expression can be explained by RNA expression. So the other 60% of protein expression cannot simply explained by RNA expression. So this suggests post-transcriptional regulation. And RNA structure-based regulation is one post-transcriptional regulation. So our lab is very interested to study the roles of RNA structure and their functions in different cellular processes. Just like any other proteins and DNAs, RNA also could fold into secondary and tertiary structure, and RNA structure will confer its function to this gene. Previous studies have shown that RNA structure could affect translation, decay, and localization, but all the previous study actually is based on the bulk cell level. So in this project, we are trying to study RNA structure at a single cell level. The aim of this project is developing a single-cell RNA structure sequencing approach to evaluate the relationship between RNA structure and their functions. Basically, the relationship between RNA structure heterogeneity and RNA functional heterogeneity. Okay. So far, single-cell technology have developed to study the transcriptome, genome, epigenome, and proteomes. So many different single-cell technology have been developed to study RNA, DNA, and proteins. But so far, there is no approach to study RNA structure at a single cell level, because current RNA structure study requires millions of cells as star material. So in this project, we're trying to study the roles of RNA structure sequence at a single cell level. We first need to develop the single cell RNA structure sequencing approach. Here is one of the rationale of RNA secondary structure probing. Basically, if we treat the cells or treat the RNA with structure probing compound, this compound will introduce a modification at a single strand area. If we do a reverse transcription under certain condition, the reverse transcription will jump through this modified position. At the same time, they will generate a mutation. So if we sequence it and calculate the mutation rate at each base, the mutation rate equals number of mutations at each base divided by total read depth of this base. So the high mutation rate will represent a single strand region, because the structure probing compound will prefer label the single strand region, or the low mutation region will represent double-strand region. So after normalization, we also call this mutation rate as structure reactivity. So higher mutation rate or higher reactivity, which represent single-strand region, or lower mutation rate or low reactivity, will represent double-strand region. So based on this mutation rate profile, or based on this structure reactivity profile, we could predict RNA secondary structure like this. Okay. To the single cell RNA structure sequence, the first step is to identify the compound, which could help us generate a high mutation rate. To this, we use tetrahedral ribozyme RNA secondary structure as a benchmark to test. Very interestingly, in all the compound and the conditions we test, we found that NN3 generate much higher mutation rate than other conditions. And for all the peak region, which is well matched to the single strand region of the tetrahedral secondary structure. So this suggests that NN3 can help us capture accurate RNA structure signal. So we use NN3 for our single cell RNA structure sequence approach. Here is the workflow. Basically, we treat the cells with NN3 and then do a cell isolation and analysis. After that, we'll do an RNA fragmentation to gently fragment the long RNA into short ones. After that, we'll do a reverse transcription jump through and template switch. After we get the cDNA, we'll prepare the library by following a normal TN5 transpose method. Here, RNA fragmentation is a critical step because if that RNA is very long, so after NN3 treatment, the long RNA will have a lot of modifications. Although the reverse transcription can jump through this modified position, but if the RNA is very long, which have a lot of modifications, they will still generate a lot of RT drop-offs. In that situation, for the long RNA, maybe it's not enough for us to calculate the RNA structure signal at a single cell level. Thus, to capture more RNAs at single cell level, we first do an RNA fragmentation to gently fragment the long RNA into shorter ones. Here is the library size of the normal single cell RNA-seq library prep. And after fragmentation, so you can see that even for DMSO and NN3, our fragmentation in the peak region is around 1K, compared with the wild type, it's 1.8K. So our library size is slightly shorter, but it's still enough to capture the RNA structure signal for at least 1 kb from the 3' end to 5' end. Especially for the longer RNA, they will help us at least capture enough RNA structure signal at the 3' end. For short RNAs, we can capture the RNA structure signal for the enough RNA structure signal for the entire RNAs. Okay. After we sequence the RNA structure signal at the single cell level, we first need to check the accuracy of our single cell RNA structure sequence approach. To this, we use 18S ribozyme RNA crystal structure as a benchmark to test. You can see clearly the AUC curve from bulk cell or from pseudobulk cells. With both of them, we can capture accurate RNA structure signal of 18S ribozyme RNA. The pseudobulk is the average of single cells. Okay, and the DMSO is a negative control. The AUC curve from each individual cells also suggests that we could capture accurate RNA structure signal for 18S ribozyme RNA. The whole AUC curve suggests that our single cell RNA structure sequence approach works. Okay. After that, we want to ask a few questions about the RNA structure signals, because we know that RNA expression have heterogeneity between different cells. So the first question we want to ask is similarly with the RNA expression heterogeneity, is whether the genes will show structural heterogeneity in individual cells. Okay. If the RNA structure also shows heterogeneity, then what are the biological reasons of RNA structure heterogeneity? Okay, if the RNA structure heterogeneity have biological reasons, meaning the RNA structure heterogeneity is not random noise. If the RNA structure heterogeneity is not random, Then, could we use RNA structure to class different cell type or different cell stage? Okay. If we could use RNA structure class different cell type or different cell stage, then could we use RNA structure as cell type, cell stage specific markers? Okay, let's check the question one by one. The first question, whether the genes shows structural heterogeneity in individual cells. To evaluate the unstructured heterogeneity at gene level, we calculate the deviation of cosine distance against pseudo bulk. Basically, the smaller distance against the pseudo bulk shows this, meaning the ion structure are more homogeneous. And the bigger distance compared with the pseudo bulk, meaning like they are heterogeneous ion structure. Obviously, 18S ribosome is one of our most homogeneous ion structure. Okay, this is agree with our previous understanding. 18S ribosome ion structure is relatively stable between different cell types. Maybe it's still hard for you to imagine what is the ion structure heterogeneity for the different genes. Here I show you two examples. One gene is RPL the DNA, which is one of our most homogeneous ion structure. You can see that this heat map. Each column represents a cell. Each row represents ion structure signal at each base of RPL the DNA. You can see for most of the positions, the ion structure signal are very consistent in different cells. So we consider this is homogeneous ion structure. Oppositely, for FKBP4, so similar, each rows represent a structure signal at each base for FKBP4. Each column represents a cell. You can see in a lot of positions, the ion structure signal are not very consistent between different cells. So this suggests that this is a heterogeneous ion structure. Those two examples also suggest that our way to analyze the ion structure heterogeneity is accurate. And then we want to know what are the functions for this heterogeneous ion structure and homogeneous ion structure. So we run a simple gene ontology for the top 25% homogeneous ion structure and top 25% heterogeneous ion structure. Very interestingly, we found that for the top 25% homogeneous ion structure, the enriched goal term are related with ribosome subunit assembly, translation, transcription. So this suggests that these genes, their structure and the very tight regulations in this biological process, or these genes, their stable structure are important for this biological process. Oppositely, for this top 25% heterogeneous ion structure, their enriched goal term are related with MRI stability and localization. So this suggests that these genes, their ion structure are under multiple different regulations. We are very happy to get this data because this suggests that ion structure heterogeneity have a biological meaningful. Okay, then we applied this single cell ion structure sequence during to the whole neuron differentiation process from human ES cells to neuron progenitor cells to immature neurons to neurons. Okay, very clearly, in human ES cells, the genes are way more homogeneous than other three time points. Here, all the genes we chose are the common expressed genes in all the 4 stages. Even these 4 stages for all the common genes, it still shows that human ES cells, their ion structure are more homogeneous. This is agree with our previous single cell ion-seq data. Human ES cells is also the most homogeneous ion expression compared with other stages. Although the ion structure signal and our previous ion-seq signal, I use different gene set, but both of them shows human ES cells are most homogeneous from both ion expression and ion structure. Okay. So from the previous slides, we know that the ion structure are really heterogeneous. Even the same genes, they have different heterogeneity at different cell types. So we want to know what are the biological reasons of this ion structure heterogeneity. We hope it's not random. Okay, to evaluate the ion structure heterogeneity, to study the reasons of RNA structure heterogeneity, we first evaluate the RNA structure heterogeneity at a region level. Because once we know that which region of the RNA structure are more heterogeneous than other regions, for example, the tree primitia is more heterogeneous than the CDS, or the CDS is more heterogeneous than the CDS. So, and later we know that we could narrow down the reasons of RNA structure heterogeneity. To this, we calculate the linear correlation between total read depth and mutant read count to evaluate RNA structure heterogeneity at the region level. For example, RPL27 at position 90. So at this dot plot, the y-axis shows number of mutations. The x-axis shows the total read depth at this base. Each dot represents a cell. You can see the correlation between number of mutations and total read depth are a very linear correlation. So this suggests that although the expression level of each base are different in different cells, but the mutant rate numbers also are linear correlation with the total read depth. This suggests that this region at different cells, they have homogeneous RNA structure signal. Oppositely, for RPL41 and position 30, you can see that the correlation between number of mutations and total read depth are not linear correlation. So this suggests that this region are heterogeneous region. And after that, we plot all the RNA structures at each region along the genes. It clearly shows that the tree primitia have way more heterogeneous RNA structure than the CDS. Because our protocol cannot capture too many RNA structure signal at 5' primitia, so 5' primitia we cannot conclude. If we only compare the tree primitia and the CDS region, we can see that the tree primitia are way more heterogeneous than the CDS region. And this will be easier to help us to narrow down the candidate reasons of RNA structure heterogeneity, because at tree primitia, there are a lot of RNA binding proteins that prefer binding the UTR region, or there are a lot of macroRNAs also binding the UTR region. Also, a lot of other environment also caused RNA structure heterogeneity at the tree primitia. So we could check these reasons one by one. First of all, because a lot of RNA binding proteins they prefer UTR region, so we first think about maybe RNA binding proteins the reasons of RNA structure heterogeneity. To evaluate the RNA binding protein contribute to the RNA structure heterogeneity, we separate 4 quantiles based on RNA structure heterogeneity. Basically, from the very homogeneous RNA structure to very heterogeneous RNA structure, and look for the RBP's target based on their RNA structure heterogeneity. For example, PAM2, you can see clearly most of PAM2's target, their RNA structure signal are more heterogeneous than others. So this suggests the RNA binding protein PAM2 binding target are enriched in more heterogeneous RNA structure. Okay. Similarly, we also separate the 4 quantiles based on RNA structure reactivity. If you still remember, the high reactivity represents single-strand structure. So if we separate the 4 quantile based on RNA structure reactivity, meaning like we separate the RNA structure signal from double strand to single strand. High reactivity represents single strand. Again, we found that PAM2 are enriched in the more single strand region. Maybe it's still a little bit difficult for you to understand. So I'll give you an example. We use PAM2 as an example. PAM2 is one of our interesting RNA binding proteins during neurogenesis. Because our single-cell RNA structure sequencing approach could help us separate. Because our single-cell RNA structure sequencing approach could help us capture RNA expression and RNA structure at the same time. So we could separate the cells based on PAM2's expression. Here it shows that we separate our cells into 2 populations. One population is PAM2 low expressed cells. One population is PAM2 high expressed cells. Okay, and then we want to check the RNA structure signal of PAM2's target RNA. Here is look like this. For the PAM2 high expressed cells, we identified more palm-tooth target, they have more single-strand region, or they have higher RNA structure reactivity. Well, in palm-tooth low expressed cells, there are less palm-tooth target. Their RNA structure signal are high reactivity. So this suggests that palm-tooth high expressed will cause higher reactivity of palm-tooth target. Okay. To validate this, we overexpress palm-tooth in human ear cells and do single-cell RNA structure sequence. You can see this heat map. Each column represents a cell. Each row represents an RNA structure signal of palm-tooth target. You can see clearly after we overexpression palm-tooth, most of palm-tooth target they will change from double strand to single strand. The yellow represents the higher reactivity, which represents the single strand region. Okay, this is clearly suggests that palm-tooth binding causes the structure change. And then we want to know, does palm-tooth overexpression or palm-tooth knockdown will cause the RNA structure heterogeneity change? So we calculate the RNA structure heterogeneity after overexpression palm-tooth. We do see that after overexpression palm-tooth, there are more RNA structure of palm-tooth target. They become more heterogeneous. Well, when we knock down palm-tooth, so more palm-tooth target, their RNA structure signal become more homogeneous. So this is agree with our previous understanding or our previous study shows that. So palm-tooth binding will cause RNA structure heterogeneity change of its target. Okay. Actually, apart from RNA binding protein binding, we also notice that even for the most homogeneous RNA structure, 18S ribosome, we also identified there are 3 regions which we labeled red color are very heterogeneous. And when we plot the regions of the RNA structures of these 3 regions, it clearly shows that these 3 regions are just localized the translation tunnel. So we think that this RNA structure signal, RNA structure heterogeneity signal may be related with translation. We cannot do the RNA translation at a single cell level, but we know that previous study shows at different cell cycle stage, they have different translation rate. So we can separate our cells into different cell cycle stage and then compare the RNA structure signal of 18S ribosome. Here it clearly shows that after we separate S phase and G2M phase, you can see this region shows big structure difference of 18S ribosome. When we zoom in, we found that these 3 regions are exactly the RNA structure heterogeneity region in this analysis. So this suggests that the different cell cycle stage caused translation rate different, which then related with RNA structure heterogeneity of 18S ribosome. Okay. After we know that there are different reasons caused RNA structure heterogeneity, they are biological reasons. So we know that the RNA structure heterogeneity is not random. Then we want to know, could we use RNA structure to class different cell type or different cell stage? Okay. What we do is that we use MOFA to class cells based on expression and structure. If we use the expression only to class different cells, we can see that we can roughly separate different time point, but the clustering is not very good quality. If we cluster the cells based on expression plus structure, we can see that the clustering is much better. And the AI value, which is used to evaluate the cluster quality, so we can see that the AI value is increased from 0.32 to 0.76. So this is a great improvement with the structure information. Although we use different genes for expression and RNA structure for class different cell types. And the structure information, which we use the commonly expressed genes in all the 4 time points. Mostly, they are very abundant genes and do not show RNA expression difference. This data clearly suggests that RNA structure signal, even their expression level do not have a difference, the RNA structure signal still could help us separate different cell type or different cell stage. This is also suggests that the RNA structure also have cell stage or cell type specific, or maybe RNA structure could serve as a cell type, cell stage markers in the future. Okay. After we know that we could use RNA structure to better class different cell types or different cell stage, then we're wondering, could we use the RNA structure as cell type, cell stage specific markers? Okay. This is simple because we can simply compare the RNA structure difference between different time points. We can see very clearly. So between human ES cells and neuroprotein cells, we have a lot of different RNA structure difference. The yellow color represents single strand region. The blue color represents double strand signal. Okay, this data shows that different time points have different RNA structure signal. To give you an example, we use RPL41 as an example. We can see that we identified two regions shows structure difference between during the whole neuron differentiation process. For position 160, we shows that this region is from high reactivity to low reactivity, which meaning this region from single strand become double strand. Oppositely, for position 310 shows that the unstructured signal from double strand becomes single strand. This is only an average unstructured signal from each time point. If we plot the unstructured signal into different cells at a different cell stage, you can see that in human ES cells, at position 160, there are more cells have higher unstructured reactivity than other time point. Well, and position 310 shows that in human ES cells, have less cells have higher unstructured signal at position 310. This suggests that these two regions are structure specific for different time point. Although there is no expression difference between all the 4 time points. So this is also suggests that some structure signal could serve as the cell type or cell stage specific markers. We are very happy to get this data, and we are dig for more target or more candidate to serve as biomarkers in different cell stage. In the future, we want to identify the cell type or disease cell type specific structure markers when we cannot simply find the expression markers. Okay. So the take-home message is that we developed an approach to accurately capture unstructured signal at a single cell level, and we found that ribosomal translation and transcription are more structurally homogeneous. This suggests that the importance of stable structure for its functions. And unstructured are more homogeneous in human ES cells than differentiating neuron cells. And the heterogeneous region are largely enriched at the UTR region compared with CDS. RNA-binding proteins, translations, and cell cycles are the reasons caused structural heterogeneity. And structural heterogeneity plus gene expression can better class different cell type or different cell stage. And structure signals could use as cell type, cell stage specific markers, even there are no difference in their gene expression. And we really welcome the collaborations and the discussion for the study of RNA structures or RNA expression or other RNA technologies. Do email me if you are interested in our project. I'm happy to take questions. Thank you.
Thank you, Dr. Wang. That was a fascinating presentation. Before we move on to the second part, a quick audience poll should be appearing on your screen now. We would like to know what field of research have you and your team been conducting? This should be a multiple choice question. You can choose 3 to 4 options. While you do that, I will hand over my time to our second speaker, Shuyun Ding. Shuyun, whenever you're ready.
Thank you for joining our webinar today. I'm Shuyun Ding, Technical Support in Vazyme. The topic of today's webinar is single-cell sequencing technology. In this webinar, we are going to first introduce single-cell sequencing, then discuss the experimental protocols and applications. Let's start with the introduction. In October 2017, the first 38 pilot project published on Human Cell Atlas mapped the 37.2 trillion cells in human body. Although human cells commonly share one set of genome, but cells are always changing in real time. The human cell map drawn by sequencing data can help us understand the type of regulation within cells, between cells, and the molecular mechanisms of differentiation and responses among tissues. Therefore, in recent years, single-cell sequencing technology became a noteworthy method. So what is single-cell sequencing? Single-cell sequencing refers to the amplification and sequencing on DNA or RNA from one single cell. Usually, it can be classified into single-cell whole genome and single-cell transcriptome sequencing. So why do we need single-cell sequencing, and why it is so important? Single-cell sequencing technology could analyze samples at single-cell level and is most suitable for the study of microvolume samples. It solves the scientific research difficulties of dealing with rare, low starting materials and difficulties to obtain information on the heterogeneity of single cells from tissue samples. So this technology could be applied in various research fields, includes pre-implantation genetic screening, circulating tumor cell detection, and nerve stem cell analyzation, etc. Moving on to part 2. In this part, I'm going to introduce the experimental protocols of single-cell whole genome and single-cell transcriptome sequencing. The experimental workflow has 3 major steps: single-cell dissociation, single-cell genome or transcriptome amplification, library construction, quality control, and sequencing. Let's begin with the first step, single-cell dissociation. Usually we need to choose different single-cell dissociation plan or protocols depends on different cell type. Most of the time we could easily find this information on website protocols.io. Usually single-cell samples can be dissociated by using the following method, such as serodilution, micromanipulation, or laser capture microdissection to isolate cells under microscope, flow cytometry to sort cells in the same type based on the markers on cell surface, and microfluidic chip, which is able to select cells by chip channels. After dissociation, cells need to be amplified. Single-cell whole genome amplification is for DNA sequencing, and single-cell transcriptome amplification is for RNA sequencing. Let's first look at single-cell whole genome amplification method. Since the low amount of starting materials cannot meet latest sequencing requirements, additional amplification step is required. But amplification is very likely to show errors and bias, so it is very important to choose suitable amplification method in order to get reliable sequencing results. The generate oligonucleotide prime PCR, MOBEC, and MDA are the 3 mainstream amplification techniques at present. COP-PCR uses partial random priming method and has the advantage in CMV detection, but with low gene coverage rate. MOBEC using multiple annealing and looping-based amplification method. It can produce relatively high output yield with reliable and reproducible results. NDA using multiple displacement amplification method. It could produce results with high gene coverage rate and accurate SNV detection. Also, this experimental workflow is fast and simple. In the next slide, I'm going to mainly focus on introduce MDA amplification method. Instead of PCR amplification, MDA rely on strand displacement amplification, using 529 DNA polymerase to amplify genomic DNA at a constant temperature. Vazyme discovered single-cell whole genome amplification kit using MDA method is applicable for unbiased whole genome amplification in about 2 hours. The size of amplified products are between 2 to 100 kb, with an average length greater than 20 kb. The qPCR primer uniformity is also great according to the quality control report. Then about single-cell whole transcriptome amplification. It was first published in 2009. In 2012, SmartSeq technology appeared, and it is able to detect full-length mRNA transcripts, but it has certain limitations. For example, it cannot efficiently transcribe sequences over 4 kb. So SmartSeq 2 technology appears in 2014, optimized the original SmartSeq technology, and become the most widely used technique at present. After that, in 2015, high-throughput dropseq technology appeared, and then 10 times genomic also appeared based on dropseq technology. As a cutting-edge technology, single-cell sequencing method was listed as one of the most noteworthy fields in journal Nature Method and Science. The major difficulties in single-cell sequencing is the low nucleic acid input amount, especially in single-cell whole transcriptome sequencing. The single-cell total RNA is about 10 picogram, and 95% of them are ribosomal RNA. mRNA only take up 1 to 3%. The minimum amount of RNA required in regular transcriptome library preparation are at least 10 nanogram, which is about 1,000 cells. Therefore, due to the extremely low nucleic acid amount in single cell, we need 100,000-fold amplification, and it is very important to choose a suitable amplification method to overcome PCR virus during the amplification. As mentioned above, SmartSeq 2 is the most widely used technology in transcriptome amplification. The reverse transcription primer oligo-DT can first attach to 3' poly A tail to avoid the reverse transcription of poly A tails. Then MMLV reverse transcriptase can recognize the cap structure at 5' end and synthesize full-length cDNA. After complete reverse transcription, the MMLV enzyme will add several additional C bases at 3' end of the new synthesized cDNA. Then TS oligoprimer will be added and let MMLV enzyme continue to act as polymerase to get double-strand cDNA. Vazyme mRNA amplification kits are based on the SmartSeq 2 method and can be used to amplify double-strand cDNA for PCR or downstream RNA library preparation. The single-cell transcriptome amplification method is applicable for different types of cells from various species, for example, different human cancer cells, mouse nerve cells, liverfish, rice, etc. After the whole genome or transcriptome amplification, library can be constructed for sequencing. For library construction, DNA first needs to be fragmented into smaller pieces. There are two different fragmentation methods, physical-based fragmentation method and enzymatic-based fragmentation method. Physical-based method is required physical sharing machine, so it has strong randomness. After fragmentation and repair, adaptive ligation, and library amplification can be performed to build library. Vazyme DNA library preparation kit included these modules and are applicable for DNA library preparation on broad DNA input range and extensive species. In addition, Kit ND608 has additional DNA damage repairing module, which is specifically designed for FFPE samples to reduce false positive rates and increase sequencing quality. If physical sharing machine is not available, we could choose enzymatic-based fragmentation method. This method fragments DNA sequence by enzyme mix, so sample wastage is low. Therefore, it is recommended when the samples are rare or precious. Vazyme enzymatic-based DNA library preparation kit has the advantages in combining the fragmentation and repair and de-entailing 3 steps into one single step, efficiently simplifies the experiment procedures, and saves a lot of time. Purification and site selection are also important to perform during library preparation to get the desired library for sequencing. The use of clean beads are one of the easiest and fastest way. DNA clean beads are commonly used for DNA or RNA library purification and library site selection, while RNA clean beads could use to concentrate RNA samples in low concentration or purified ribosomal and depleted RNA in the RNA enrichment step. Library quality control is also a very important step after library preparation were completed. Qubit could be used to measure library concentration, while analyzer could detect library fragment distribution, and qPCR could also be used for accurate quantification of samples concentration. Moving on to part 3, I'm going to introduce some applications on single-cell sequencing. This article is a research in single-cell RNA-seq in mouse embryonic development. Since cells are highly heterogeneous during mammalian development, research needs to be done at single-cell level. This research used single-cell RNA-seq technology to sequence more than 2 million cells, detected more than 500 cell types, and systematically mapped the cell differentiation pathway and the changes in gene expression. These information are very important for embryonic development study and organ formation in mammals, including humans. Single-cell sequencing technology is also significant in the study of tumor immune system, for example, the functional state of T cell. In this study, by using single-cell full-length transcriptome sequencing technology, the relationship between the heterogeneity of colorectal cancer patient and T cell were analyzed, revealed various characteristics of the cancer T cell, making the discovery of new target genes become possible. So far, it is the most comprehensive and novel single-cell study on T cell in tumor microenvironment in the world. That's all. Thank you for your attention.
All right, thanks for the two of you giving us the presentation. Now we have a few questions for the speakers. Now, Dr. Wang, just recently you have tested out our VAZ DNA clean beads N411. How do you feel about this particular reagent?
Yes, so I compared the beads, the magnetic beads from Vazyme and whether they use impure beads. So the overall performance are very similar. So the library size less than 100 pp all have been removed, and the library size around 200 pp and above be kept. The yield efficiency compare both of the two beads, they are very similar. So I guess it is a very compatible result. I think it's good to use as other brands.
Okay, so would you say the performance of our N411 would meet your research needs?
Yes, it's absolutely. So when I compare, although I only do one, but the performance is like 10% better even than whether they use the impure beads. I think it's really enough. The quality is enough for us to do it.
Okay, thank you. Okay, so we have a few other questions for Christina, which is Shu Yuanding, who has given the presentation earlier before. The first question is, what type of sample type could be used with Vazyme single-cell RNA amplification kit?
Vazyme single-cell RNA amplification kit N711 and N712 use oligo-DC primer to amplify RNA samples with poly-A cells. It is applicable for mammalian cells or other eukaryotic cells without cell walls. It is also suitable for 10 picogram to 10 nanogram of purified total RNA with poly-A sequence. However, it is not applicable for prokaryotic cells or fixed cells.
Okay, so the second question is, what should be checked during the single-cell sample preparation stage?
Since RNA can be seriously degraded in dead cells,
Cell viability should be detected by trip and blue staining or other methods to avoid library preparation failure. Also, please make sure the study material is inside certain range. For example, 1 to 1,000 cells per library for N711 or 1 to 500 cells per library for N712. Excessive cells will not increase the output but may cause inhibition on the whole reaction.
Okay, so we have a last question for you. Can multiple single-cell library samples be put together for sequencing?
Yes, like the common polling method in sequencing step, multiple single-cell library samples with different acceptors can be put together for sequencing.
Okay, thank you. So now we have all the time for today, and I hope to see you all next session. Okay, thank you. Bye-bye.